Okay, just to pick up where we left off. So this is the summary for glycolysis and fermentation. So glycolysis on its own produces too much NADH, and it's got to be recycled to oxidized NAD+. And the way this works is by using, pyruvate or other organic molecules derived from pyruvate to function as electron acceptors. So these processes are important industrially for some of them and in the food industry. Yogurt, cheese, this kind of stuff, they are bacterial fermentation products. And they occur, so fermentation occurs in anaerobic conditions, no oxygen, or when sugar is plentiful, because metabolically these pathways are very, very simple. So if sugar is in a huge excess, like, I don't know, 10 grams per liter or something in a medium, then bacteria, even if oxygen is present, will ferment. So that's why, for example, the stuff we saw about identification of Staphylococcus aureus in Chapman medium, there's a high concentration of mannitol. Those bacteria will ferment the mannitol, even though oxygen is present, because there's so much mannitol around. Just use a lot and ferment it and throw out the fermentation products. Okay, so occurs in the absence of oxygen, but also when you have a large excess of sugar. Okay, so if bacteria are growing in oxygen with a relatively small amount of sugar or peptones or some other carbon source, then if they can do it, they will produce energy by aerobic respiration. And there are three main stages here. So the first is glycolysis, breaking down glucose to pyruvate, producing NADH and some ATP. Then pyruvate will be converted to acetyl coenzyme A, combined with oxaloacetate, and this 6-carbon citrate will go around the Krebs cycle, and this will produce some ATP and a ton of reduced NADH and FADH. So that's a second step, the Krebs cycle. All of the carbon has been oxidized to CO2. A lot of NAD+ has been reduced to NADH. And then the third step is to get rid of the electrons on NADH by transferring them onto an electron acceptor, which is going to be oxygen. And during this process, this can generate a proton gradient, which can be used to produce ATP. So this is the last step, electron transport and oxidative phosphorylation. But once again, you're going to see this, I guess, in detail in biochemistry. So I don't want to really go through all the steps here. But just for example, in E. coli, they have a simple electron transport chain in the plasma membrane. So it starts out with lots of NADH in a cytosol, and the imaginatively named NADH dehydrogenase will remove the electrons from this NADH and transfer them onto coenzyme Q, or ubiquinol. Ubiquinone, in fact. So what is that? Okay, ubiquinone is this organic molecule here. So you've got two ketone groups here, which will be reduced to alcohol groups. Okay, this will become OH, OH. And this is the reduced form of ubiquinol. So what is happening here is that the electrons and two protons are going to be transferred to ubiquinone, which picks up 2 electrons and 2 protons from the cytosolic face of the membrane. And then, okay, so this is a kind of hydrophobic chain here, so it's soluble in the plasma membrane, so it diffuses in the membrane. It's going to diffuse along over here to this cytochrome oxidase complex, and the electrons are going to be transferred onto a series of cytochromes, and the protons will be liberated on the periplasmic face of the membrane. So the ubiquinol cycle picks up protons from the cytosol and delivers them into the periplasm. So this is one mechanism to generate a proton gradient. So that's a very important part. And the other thing that happens afterwards is that, okay, the cytochrome oxidase complex is finally going to transfer these electrons onto water. And of course, no, onto dioxygen and form water. Okay, so oxygen gas is going to be combined with protons, and the electrons come from here to produce a molecule of water. So once again, this uses up protons on the cytosolic face of the membrane and also contributes to the proton gradient. Now, E. coli have two electron transport chains, the BO branch and the BD branch. So the BO branch is over here, and it functions when there is a lot of oxygen present. So the cytochrome oxidase complex here has a low affinity for oxygen, but when it works, when high concentrations of oxygen are present, it allows the translocation of more protons. So it's more efficient in terms of generating the proton motive force. In low oxygen conditions, the BO branch won't work. And this is where the BD branch comes in useful. So this has high affinity for oxygen, but doesn't translocate so many protons. But it's still good. So you have these two branches of the electron transport chain that work under high and low oxygen conditions. Okay. Oh yeah. The other thing is cytochromes. What are cytochromes? Okay. So cytochromes are small proteins that have a heme group, and the ion in here is what's going to flip from the ferrous to the ferric form. So iron 2 to iron 3, these are involved in the electron transport. So each of these, like little symbols here, B558, B595, D, these are different cytochromes, and the electrons are being transferred from one to another. Okay, that's why iron is so essential for bacteria. Well, not just in this process, but in a lot of processes where you have electron transport. A lot of redox reactions. Now, the proton, so the last step is using the proton gradient to produce ATP. And this is the proton ATPase, which is in the plasma membrane. So this part, the head group, is in the cytosol. The proton gradient, more protons outside than inside, so the protons run down the gradient. This causes this head group to rotate and allows the formation of ATP. So the proton ATPase is the last step. And when this is working, then if you've got a lot of oxygen and the BO branch is working, then theoretically this can allow bacteria to produce 38 molecules of ATP per glucose. You know, this is way more efficient than glycolysis and fermentation. Now, when oxygen is not present, some bacteria can perform what is called anaerobic respiration. It sounds like it's impossible, right? Respiration is breathing, so how can you breathe if you've got no oxygen? However, respiration here means electron transport and generation of ATP. And in this case, in anaerobic respiration, the electron transport chain is going to end up transferring the electrons not onto oxygen, but onto nitrate. And in this particular example, Paracoccus denitrificans, the end result is going to be the formation of nitrogen gas, N2. So why is this possible? Okay, it's because of the redox potential of the donors. So in the beginning, all these electrons are on reduced NADH, okay, in the cytosol over here. So you've got a redox potential of -3 volts. For this half reaction here, nitrate to nitrite, the redox potential is +4 volts. So your overall redox potential is +7. Delta G, very negative. So you can easily get a transfer of electrons from NADH onto nitrate, which will be reduced to nitrite, and then from nitrite, which can be reduced to nitrogen gas. So these are good. So nitrate is a very good electron acceptor if it's present. Now, that's not the only, so nitrate is not the only possible electron acceptor for anaerobic respiration. Sulfate can be used, iron, ferric iron can be reduced to ferrous iron, and even these weird mineral salts here, arsenate and selenate, can be used as terminal electron acceptors if they're present in large amounts. So generally what bacteria will do is they will use the best electron acceptor first, which is going to be oxygen. Then when oxygen runs out, use the next best, which will be nitrate. And then apparently manganese 4 is the best, then iron, then sulfate. And then finally, carbon dioxide can be used as an electron acceptor. So this occurs in, underground aquifers when there is an input of organic carbon at the beginning, because all of these bacteria are gaining energy by, for example, let's say that. Let's just keep it at that stage now. So the energy comes in the form of organic carbon. It's going to be metabolized by glycolysis, produce NADH, and these different minerals will act as terminal electron acceptors for anaerobic respiration. And I think the kind of exception is methane production here. So the electron donor is going to be either, it's going to be hydrogen gas at some point. Either produced as a fermentation product by other bacteria, or it can be produced by minerals. Okay, so that's anaerobic respiration. Same as respiration, except another electron acceptor is used. So far we've looked at organic carbon as the source of electrons and as the basis of the energy metabolism. Okay. Now we're going to switch to other sources of metabolic energy. So this is going to be chemolithotrophs, using an inorganic chemical reaction as the source of energy. And there are 3 types: hydrogen oxidizers, nitrifying bacteria, and sulfur oxidizers. So hydrogen oxidizers, well, that's no problem, because like dihydrogen gas, it's like a really fantastic energy source. Think about hydrogen fuel cells. If you burn, they can generate energy. You drive your car with them. So it's not surprising that dihydrogen gas can be used as a source of energy by bacteria. What is more surprising is these guys, the nitrifying bacteria and the sulfur oxidizers, because they will be performing kind of like the opposite of anaerobic respiration. So here, the reduced forms of nitrogen or sulfur will be the electron donors, and that can only work if they have a really good electron acceptor. So that's going to be oxygen, mostly. Okay, so anaerobic respiration, nitrate, sulfate can be the electron acceptor. For chemolithotrophy, ammonium hydrogen sulfide will be the electron donor, and oxygen is the electron acceptor. Okay, so the tricky thing, so it's the presence or absence of oxygen which determines which way round it's going to go. Okay, whether the nitrogen and the sulfur compounds are going to be electron donors or acceptors. So that's generally how it works. So how do these organisms generate their metabolic energy? So for hydrogen oxidizers, there are aerobic hydrogen oxidizers. So alkyligenase hydrogenophaga eats hydrogen, aquifex makes water. So electron donor is hydrogen, electron acceptor is oxygen, produces water. Methanogens are also hydrogen oxidizers. So here the electron acceptor is carbon dioxide, and this produces methane. So these bacteria are going to be autotrophs, right? They're getting metabolic energy in a very difficult way, just from some kind of mineral source. Probably there's no organic carbon around here. They're growing just on a simple chemical reaction. Now, hydrogen gas is such a good electron donor that it can actually reduce NAD plus directly and form NADH. So look at the redox potential here. Okay, it's an even better electron donor than NADH. So, you know, an enzyme can just, you know, transfer the electrons onto NAD plus. It's a favorable reaction. And once you've got that, then ATP can be produced by the electron transport chain, and the electrons finally go on to the electron acceptor, which will be oxygen. Easy. So that works pretty well for these aerobic hydrogen oxidizers. In methanogens, their metabolic pathways are very complicated because carbon dioxide is such a poor electron acceptor. But nevertheless, they manage to perform these reactions and extract some kind of metabolic energy from the process. Nitrifying bacteria. Okay. Nitrosomonas will use ammonium as the electron donor and oxidize it to nitrite using oxygen as the electron acceptor, and then Nitrobacter will take the nitrite and oxidize it to nitrate. So these are called nitrifying bacteria because in the soil or in water treatment plants, in the presence of oxygen, they transform all the toxic ammonia compounds into nitrate, which can be assimilated by plants. Now, what's the point of this here? Okay, yeah, look. If you're living by oxidizing nitrite to nitrate, you know, nitrite is a pretty good electron acceptor, in fact. All right? So the redox potential here is plus 0.42. And if you're going to transfer electrons from nitrite to NAD+, this is never going to work directly because it's totally the wrong way round. Everybody understand why this is the case here? I hope so. Yeah, look. Here is nitrate to nitrite. And here is NAD plus to NADH. Normally, this has got to be the electron donor, and the nitrate will be the electron acceptor here. Here we've got bacteria that are trying to produce NADH by taking electrons from nitrite. No. Yeah, that's right. How can that possibly work? Okay, well, how it works is that this can be used to generate proton gradient. Okay, just by transferring the electron from nitrite and then electron transport chain to put it onto oxygen with a cytochrome oxidase. And that's enough to produce a proton gradient. Then what you can do is the electron transport chain that goes from NADH to NAD plus is going to be reversed. Why can it be reversed? Because if you've got a very high proton gradient, then this will be associated with a potential difference. That potential difference can be high enough to push this reaction to go. So that can work. It's amazing, but it can work. Sulfur oxidizers, they do something interesting as well. Okay, so there are aerobic sulfur oxidizers that are going to take reduced forms of sulfur and molecular sulfur and oxidize them to sulfuric acid. And that is a kind of a very problematic process, especially in old iron mines, okay, because you've got a lot of old mineral iron and sulfur compounds that are open to the air, and these bacteria need to use up a lot of these mineral compounds to generate energy. So in the east of the United States, these bacteria will be producing millions of tons of sulfuric acid, and it will all seep out into the rivers, into the water. And the solution to this is actually to block up the mines, because the whole process requires oxygen. So if you block up the old disused mines, then it will prevent the problem of acid seepage, which is caused by these bacteria. Now, the other type of sulfur oxidizers, thiobacillus denitrificans, they can generate energy anaerobically by using hydrogen sulfide as the electron donor and nitrate as the electron acceptor. Okay, because if you remember in the order of who's the best electron acceptor, you've got oxygen, nitrate, then sulfate. Okay, so reduced sulfur compounds can give up their electrons to nitrate. What do I have to say about this? So you've got like two molecular processes biochemical processes going on here. So you'll have an enzyme which is going to remove the electrons from this reduced forms of sulfur and then use them to produce proton motive force and make NADH by reverse electron transport. And there's a cute bit of biochemistry here that allows these sulfur oxidizers to generate ATP directly, because sulfite can combine with adenosine monophosphate to give you adenosine phosphosulfite. And then these can combine with anorganic phosphate to give you ADP. And 2 ADP gives 1 AMP and 1 ATP. So there's a kind of direct combination of the redox reaction and phosphorylation here. So this is very, very simple. This might be much easier than oxidative phosphorylation. Okay, yeah, so let's see if you understand this electron donor, electron acceptor stuff. Which of the following electron transfers are going to work or can produce energy? Okay, everybody thought about this? It's useful if you've got the scale with, you know, the different redox potentials for the half reactions. It's the only way you can work, you can figure it out. Is everybody ready to say something about this? Okay, glucose as the electron donor, nitrate as the electron acceptor. Does this work? Can it work? Who says yes? One person, two people. Who says no? No way, you're kidding me. Plenty of people. The answer is yes. The answer is yes. This is the definition of anaerobic respiration, right? This is the overall process. So, yeah, this works. Glucose as the electron donor, carbon dioxide as the electron acceptor. Who says this can work? One person, two people, three people, four people. Whoa, it's got a kind of snowball effect going here. Who says no, can't work? One person, you're right, can't work, okay? Carbon dioxide is the worst electron acceptor, okay? Nitrite as electron donor, carbon dioxide as electron acceptor. Oh, damn, I gave the answer right there. Can it work? No, can't work. Carbon dioxide is the worst. It's the pits, man. Pits of the earth. Okay, so only hydrogen can work in this process, okay? Because carbon dioxide is just so bad. You need like the really best electron donor you can possibly get. Otherwise, it's never going to work. Okay, how about this one? Ammonium as an electron donor, sulfate as an electron acceptor. Okay, who says yes? Who says no? A few people. Yeah, the no's are right. Okay, because you know you just have to remember who's the best electron acceptor here. Oxygen, nitrate, sulfate, then carbon dioxide. So nitrogen compounds are better electron acceptors. And if you've got a reduced form as the donor, you're going to have to find a better electron acceptor than the nitrate, than its own like oxidized form. And sulfate, that cannot work because sulfate is a worse electron acceptor than nitrate. Not so good. Only one electron acceptor better than nitrate, which is oxygen, yes. Okay, so the nitrogen oxidizers will always require oxygen. Okay, there's one exception which we'll see in a minute. And then this one, okay, will this work? Molecular hydrogen as donor, nitrate as the electron acceptor. Who says yes? Yeah, you're right. Yeah, so really good electron donor, really good electron acceptor. This will work. Good. Okay, so now we've got all the different kind of transformations that can occur. going to think a little bit about the carbon and nitrogen cycles. Carbon cycle first, and what role do microbes play. So if we just think about, us, we're quite used to this oxic part. This is where we live in an environment that has oxygen. So, plants will fix carbon, produce organic matter which we eat, and we will metabolize it by respiration back to CO2. And, so microbes maybe don't play such a big role in this. Apart from perhaps, 50% of the aerobic or oxygenic photosynthesis in the world is by microbes. So it's going to be photosynthetic bacteria, cyanobacteria in the sea, or other well, not necessarily cyanobacteria, but algae, diatoms, unicellular algae, photosynthetic microbes. So that's half of the carbon fixation in the whole on the planet. Okay, is in the ocean. Now, what about the anoxic side? Okay, so you've got autotrophs. So these kind of like nitrosomonas, these chemolithotrophs, which are also fixing carbon dioxide. They can also produce organic matter, but this is about 10,000 times less quantitatively. So oxygenic photosynthesis is, the big source of organic carbon on the planet. Then, okay, what am I doing here? Yeah, okay. Then using up this organic matter, okay, respiration, we've seen that, and then anaerobic respiration and fermentation among bacteria. And the other thing that's happening is methanogenesis. So hydrogen gas here, is a fermentation product from mixed acid fermentations. And it can be combined with CO2 to produce methane. And some of this methane can be oxidized back to CO2. Two things I need to say about this. One of them is, okay, shift the clock back about 3 billion years ago. What kind of processes are we going to see? How is our carbon cycle going to look 3 billion years ago? Okay, I'm going to. So, which bits are going to be absent? The blue bits or the red bits? Blue bits. 3 billion years ago, before the great oxidation event, oxygenic photosynthesis had not yet evolved. There's no molecular oxygen in the atmosphere. So all of this part didn't exist, 3 billion years ago. So 3 billion years ago, we've got, the carbon cycle is running like this. So the earliest microbes have, this kind of carbon metabolism, or had this kind of carbon metabolism. Some autotrophs, chemolithotrophy, anaerobic respiration, fermentation, and methanogenesis. These are going to be the most ancient types of metabolism. Now, the other things I want to talk about are, okay, the use of organic matter by different types of microbes and methanogens, and whether they are a serious threat for global warming. Okay, so for the degradation of organic carbon, now most microorganisms can use simple sugars, peptones, fatty acids, purine, pyrimidine bases as forms of carbon. They're easy to metabolize. But there are some forms of carbon that are difficult to metabolize, okay? So cellulose, for example. Not all bacteria can degrade cellulose. It depends on the scission of this beta 1 to 4 bond. So only some bacteria have the enzymatic equipment necessary for breaking down cellulose. So ruminococcus, bacteria that are commensal bacteria in the guts of ruminants and in other herbivores also have other commensal bacteria that can break down cellulose. So these bacteria will be breaking down cellulose, producing fermentation products like acetate, lactic acid, which are then absorbed by the gut of the animal. So that's how horses, cows can get some energy out of eating grass. Okay, lignin. This is the polymer in wood. Extremely difficult to break down. Okay, it can only be broken down in the presence of oxygen. There are no anaerobic pathways to break down lignin. So, wood can only be recycled into or oxidized into carbon dioxide in the presence of oxygen. So if wood is buried in an anoxic environment, it can't be fermented. And of course, it will eventually turn into coal. And mostly this is performed by molds, okay, not bacteria. So, okay, there are apparently some lignin degrading bacteria, but mostly it's done by molds, by champignons. Okay, and last one is, hydrocarbons. All of the synthetic plastics that we've been producing, they are not natural molecules. So, of course, they are very difficult to degrade by microorganisms. But there are bacteria that have been found that are able to degrade plastics. And people have found them by looking at sites where there's a lot of pollution in the ground and trying to cultivate bacteria that can grow just with ground up plastic as the carbon source. So this is one of the ideas to how can we get rid of all the plastic pollution in the world, is to try and find bacteria that could degrade them and then perhaps use them in fermenters. Put plastics in the fermenter and we'll get methane out of it, and we'll burn it to heat the home or something like that. It'd be cleaner than just burning the plastic. So mostly these degradation pathways will require oxygen. Okay? So the second thing I want to say about the carbon cycle and bacteria is methane. What's the role of methane? So methanogens are able to use carbon dioxide as an electron acceptor because they have access to a great electron donor, which is hydrogen gas. So overall, this process will work because, your delta V here is in about, 0.2 volts. Really pathetic, really very, very small potential difference here, but at least it's going in the right direction. So they can actually, live on just carbon dioxide and hydrogen gas. So methanogens are archaebacteria. Some of them live in ruminant guts. Others live in marshes, so methane known as marsh gas. And there are a lot which live in oceanic sediments off the coast. And in northern latitudes, where it's very cold, the methane is associated with ice crystals in methane clathrates. And there are large amounts of these methane clathrates down at the bottom of the ocean, up in the Arctic. And the big problem is if the temperature goes up by a few degrees, then all of the methane that's sequestered under the ocean here is going to be released. And that could be a really big problem because methane is 25 to 30 times more powerful than carbon dioxide as a greenhouse gas. So this is going to be one of the potential tipping points where if the temperature rises enough to release all these methane class rates, then it's going to exacerbate the problem and accelerate the warming. So of course, methane has a much shorter half-life in the atmosphere than carbon dioxide because it's relatively quickly oxidized to CO2. But still, it's not the kind of thing that you want to happen, right? So this is something that may occur. Well, I worry that it's going to happen actually sooner than we think. However, it's not all bad news. It may never happen because liberation of methane can actually provide a food source to other bacteria, which are methanotrophic. Okay, so there are bacteria that can use methane in an environment that is, slightly anoxic, but maybe has some oxygen present. So once again, it's an archaebacteria, are methanotrophs, and they often live in symbiosis with other bacteria. So what these methanotrophs are going to do is remove the electrons from methane to produce carbon dioxide, and then somehow these electrons have got to find a good electron acceptor. And by good, I mean anything better than CO2. So oxygen, nitrate, and even sulfate is a better electron acceptor than carbon dioxide. So the transfer of electrons from methane to sulfate, that can work, okay? In energetic terms, it's favorable. But archaebacteria, these methanotrophs, don't have the enzymes required for sulfur reduction. So they live in symbiosis with sulfur-reducing bacteria, which I believe are gram-negative proteobacteria. And what's kind of interesting about this was described in 2015, these methanotroph sulfur-reducing bacteria consortia, is that they're joined together by these type 4 pili that are produced by the sulfur-reducing bacteria that make contacts with the archaebacteria, the methanotrophs, and these are wires. These form kind of wires that transfer the electrons from the methanotroph to the sulfur-reducing bacterium. So normally the electron transport chain is a bunch of, integral membrane proteins in the plasma membrane. And here, in this case, it's actually, a kind of physical contact between the bacteria. So that's kind of interesting. So if these bacteria start to grow a lot, once the methane class rates liberate the methane, then perhaps we won't see a large release of methane gas, because if they'll be able to grow, and what we'll get is, okay, we'll be transformed to CO2. Still not great, but it's not so bad as having a huge release of methane into the atmosphere. So we might just get, carbon dioxide and hydrogen sulfide produced. Okay, that's carbon cycle. The nitrogen cycle. Now, there are a lot of transformations of nitrogen that are possible. Okay, so we've seen anaerobic respiration, and we've seen nitrogen oxidizers, and bacteria also need to assimilate nitrogen to, use it to make amino acids. And this diagram here is trying to summarize all of these processes at the same time. Now the reason I put this up here is because I think, well, the person who wrote this textbook did a pretty good job, I think, of using different colors for different types of metabolism. Okay. So these white arrows, okay, you've got nitrate going to nitrite, going to ammonium, and you've got the same thing, ammonium going to nitrite, going to nitrate. So one of these is chemolithotrophy, and one of them is anaerobic respiration. Okay, which is which? So nitrate going to nitrite. Which one is that? Chemolithotrophy or anaerobic respiration? Okay. It's anaerobic respiration because the nitrate is the electron acceptor and it's going from an oxidized form to a more reduced form. Okay? So that's what these purple lines mean. Okay? These purple lines are oxidized nitrogen being used as an electron acceptor in anaerobic respiration. No oxygen present, nitrate used as the electron acceptor. So that's why the arrows are going in this direction. Now the opposite process occurs where you have reduced nitrogen compounds, but you have oxygen present. So the process goes in the other direction because nitrogen compounds, they're good electron acceptors, but the best is oxygen, right? So when oxygen is present, these arrows go the other way around. And that means that this is happening in different environments and different bacteria are performing these different processes. Okay, you don't have the same bacterium doing the same reaction in both directions. So the reason you've got the arrows going in both directions is because basically oxygen is present or not. Okay? Yeah. So the kind of blue and pink arrows here, these ones that go to organic nitrogen anyway, these are, this is production of, fixed incorporation or assimilation of inorganic nitrogen into organic nitrogen. That's what this represents. Okay, at least from dinitrogen gas and assimilation of nitrate into organic nitrogen. Okay, this one, I don't really know what that is. This is, if you've got, this is getting rid of organic nitrogen. That is, something has died, it's released a lot of protein, bacteria taking up this protein and using it as a form, as a source of energy. So in fact, this will generate ammonium as a waste product if peptones are being used as the main source of carbon for energy production. So the carbon will all be, oxidized or fermented to something, and the amine groups that are on the amino acids will just be excreted as ammonium. So that's where these ammonium compounds come from for the beginning of these processes. And the last thing that exists here is this weird thing where there are some bacteria which can combine ammonium and nitrite to produce nitrogen gas. So this is called the Anamox process, which stands for anaerobic ammonium oxidation. So this is kind of a weird process because the electron donor, so this is, another chemolithotrophic process here. So it's bacteria getting energy from an inorganic reaction. So the electron donor is ammonium, the electron acceptor is nitrite, and they combine to form, neutral nitrogen gas. So this is a weird process. It was only discovered 20 years ago, in 1999, and it's performed by bacteria which are part of the planktomycetes group. So why does anybody care about the Anaerobic process? Well, in water treatment plants, you have to try and get rid of the nitrogen compounds before they can come back into the water supply. So mostly this is done by using denitrifying bacteria. But first, all of the ammonium compounds have got to be fully oxidized to nitrate. So this means that in the water treatment plant, you've got to bubble air through the slurry at some point to make sure that the nitrifying bacteria can oxidize everything up to ammonium, to nitrate. And then in the next step, denitrifying bacteria will use this nitrate as an electron acceptor in anaerobic respiration and release nitrogen gas. So the wastewater, the sewage waste has been denitrified. Now, the Anaerobic process may be more energy efficient because it's anaerobic. You don't need molecular oxygen to do this. So you don't need to stir the whole thing and you don't need to run air through the system. So that's the interest of the Anaerobic process. So for here, this is complicated, all right? And the most difficult thing, or the thing that I want you to be able to understand, is why the arrows can run in both directions. What is the purpose for the bacteria of each of these type of reactions, okay? So you have to be able to tell whether the nitrogen compound being used is the electron donor, the electron acceptor, or whether it's being assimilated to make amino acids. So very different processes. And that's why you've got arrows going all over the place. All right. Okay, so we have to survive another 30 minutes if we can make it. Probably won't take 30 minutes. Okay. Now, the last thing we have to think about is photosynthesis, phototrophs. So the point of this slide here is to show you that there are many different types of photosynthetic bacteria. So there are photosynthetic archaea, for example, Halobacterium, and there are many types of photosynthetic eubacteria. But there are only really two different types of basic photosynthetic process. So all of the eubacteria work with bacteria chlorophyll and either reaction center 1 or reaction center 2, or both of them, whereas Halobacterium has a totally different light capturing pigment. Okay, so we've got two basic systems for light capture. So two independent origins of photosynthesis in bacteria. One in the archaea and one in the eubacteria. But so, okay, it's pretty clear who invented bacteriorhodopsin, but who invented or where did bacteria chlorophyll evolve? And it's not an easy question at all because there are a whole bunch of different photosynthetic eubacteria, and they are not very closely phylogenetically related. Most photosynthetic bacteria are classical gram-negatives, but there are some gram-positive photosynthetic bacteria, and chloroflexi are in this kind of like a kind of unusual group, kind of a little bit further away from classical gram-positives and gram-negatives. One thing that is clear is that, okay, it's not very likely. Okay, so the question here is, okay, you've got two hypotheses to explain the distribution of bacteria chlorophyll and photosynthesis among these different groups of bacteria. One is the ancestor of all these different bacteria was photosynthetic, and all of the non-photosynthetic bacteria just lost photosynthesis. That's hypothesis number one. What's the other hypothesis? Nobody know? I think it's probably written on the next slide. Okay. What else? Okay. What the other thing is? Oh well, they all evolved photosynthesis independently. You might say that. Well, why is that definitely false? Why can we exclude that as an explanation? Yeah. Yes, and they are all quite similar. Okay, so the genes show that there was a common origin. Okay, whereas, so you can't say, okay, we've evolved exactly the same genes involved in photosynthesis in the chloroflexi, and we evolved, you know, just by convergent evolution, exactly the same genes in all these other very diverse types of bacteria. It's just incredibly unlikely. So that's why we say, okay, maybe the ancestor had this set of genes, and then all the other descendants, they inherited them, but most other bacteria just lost them. But also relatively unlikely, because you're thinking, you know, why would all the other bacteria just lose them so much? It seems like not a likely thing to happen. So if bacteria aren't losing genes, okay, if the non-photosynthetic bacteria are not losing genes, what could be happening to the photosynthetic bacteria? The non-photosynthetic bacteria do not have genes for photosynthesis. The photosynthetic bacteria have them. So maybe they got them, maybe they gained them, right? So this is the idea for, well, is that in eubacteria, there will be some kind of ancestor that was the first photosynthetic bacterium, developed photosynthesis based on capture of light by bacteria chlorophyll, and then this system was transferred to other groups of bacteria. And that explains, you know, why you've got these diverse types of bacteria that are photosynthetic. So its lateral gene transfer is considered to be the most likely explanation for this distribution of photosynthesis. Okay, so let's go back to Halobacterium here and find out how it works. So the photosynthetic system is based on bacteria rhodopsin. So this is a 7 transmembrane helix protein with a pigment molecule on the inside. And basically, when light hits this, then it acts as a proton pump. And it will transport one proton from the cytosolic face of the membrane to the outside of the membrane every time it absorbs a photon. So it's a light-activated proton pump. And this is just helping the bacterium to generate proton motive force. So there's no electron donor, no electron acceptor. Just generates a proton gradient, and then this can be used to help the bacterium produce ATP with the proton ATPase. Okay, so it's giving a kind of energy boost. Okay, so for all the diverse groups of photosynthetic bacteria, okay, we've got cyanobacteria. That's maybe, okay, it's the most complicated photosynthetic system, but it's the simplest one to understand. They work just like green plants, okay? They perform oxygenic photosynthesis. Okay, so kind of have an idea what is happening with these guys. But with the other photosynthetic bacteria, you find that they either have only photosystem 1 or only photosystem 2. Cyanobacteria, they are the only photosynthetic bacteria that have both reaction centers. And this is required in order to extract electrons from water. Okay, so you need both of them to perform ordinary oxygenic photosynthesis that occurs in green plants, it occurs in cyanobacteria. So what on earth use is it going to be if you've only got half of this system? Okay, so that's what I want to look at now in chlorobi and in purple non-sulfur bacteria. So chlorobi are green sulfur bacteria. So they have a type 1 reaction center. So light is harvested by an extracellular structure called the chlorosome, and a typical genus of chlorobi is chlorobium. So what is happening here? Okay, the electron donor is hydrogen sulfide or thiosulfite. So this is going to provide electrons here, and this electron is going to be transferred after the reaction center is going to boost the energy, the reduction potential of this electron, so that it can be transferred onto NAD and produce NADH. So if you remember, for nitrogen oxidizing bacteria and sulfur oxidizing bacteria, the redox potential of hydrogen sulfide is not a good enough electron donor to reduce NADH directly. Whereas in these bacteria, it can work because the redox potential of the electrons that come from the reduced sulfur compounds is boosted by the reaction center of the photosynthetic material here. And this allows bacteria to generate NADH. Great. So that's the use of photosynthesis for these bacteria. It allows them to generate NADH directly. So it's much more efficient than just ordinary sulfur oxidation. And also, this can occur in an anaerobic environment. Okay, so it's underwater, a little bit, doesn't have very much oxygen. So normal sulfur-based chemolithotrophy is not going to work very well. Whereas here it's going to work. Okay. And okay, reaction center type 1 is considered to be the ancestral form of the two reaction centers. RC1 and RC2 that are found in cyanobacteria are paralogs. Okay, so they are sets of homologous genes that perform slightly different functions. The ancestral form is considered to be RC1. So this is the kind of thing where you think the ancestral type of photosynthesis was probably doing something like this. allowing chemolithotrophy to be a little bit more efficient than it otherwise would be. So purple non-sulfur bacteria, what is happening here? Okay, so they have only a type 2 reaction center. They have another kind of light harvesting complex to, make light capture more efficient. An example is Rhodobacter. Now here there is no electron donor. All that's happening, the only thing that's happening is that light is going to activate an electron here at the photosynthetic reaction center, and because its redox potential has been increased by absorbing the photon, then it can be just take a turn on an electron transport chain that involves ubiquinone, ubiquinol. So if you remember, ubiquinone, when it's reduced, picks up electrons and it picks up protons from the cytoplasmic face of the membrane. When it gives up the electrons, it gives up the protons on the extracellular face of the membrane. So just by having the quinone cycle here, this allows photosynthesis to generate a proton gradient. And the bacteria can use this to make ATP. So it's the same kind of thing as we saw for Halobacterium. So purple non-sulfur bacteria, they don't need sulfur, they don't need an electron donor. They probably are picking up, I think they're basically organoheterotrophs, purple bacteria. So they're using organic carbon for their, to produce their cell constituents. And the photosynthesis is just grafted on top to give them a little bit more efficient energy use. And once again, they are in anaerobic environments. Cyanobacteria, okay, this is the same thing as happens in green plants. So what is great about this is that you've got the association of type 1 and type 2 reaction centers, and this is what allows electrons to be extracted from water. This is, almost chemically impossible, because we spent all of our time saying, oxygen is the best electron acceptor, best you can get, which also means that water is going to be the absolute worst electron donor, right? Because it's so somehow cyanobacteria managed to evolve this, and they've done it. They've hit the jackpot with this, because they can generate any reducing equivalents. They've got the quinone cycle here, so they can produce a proton gradient and generate ATP. So they can generate all their energy needs just from light and water, a ubiquitous electron donor. So, that's why the cyanobacteria are by far the most abundant and the most diverse type of photosynthetic bacteria. They're present in fresh water, several species in salt water. What to say about this, except just to remind everybody that this was an exceptionally important event in the history of the planet Earth. Changed the world forever, because without this process, there is no way for atmospheric oxygen to accumulate, because oxygen is just too reactive. So this process was absolutely essential for accumulating atmospheric oxygen, forming the ozone layer, allowing the surface of the oceans and the terrestrial environment to become habitable. And it also seems to have been a necessary step in the evolution of eukaryotes. So if this quite amazing piece of biochemistry had not evolved, then we just definitely would not be here. Okay, so the summary for phototrophs is, okay, you have to think about what is the source of electrons, what is the basic type of metabolism of these different phototrophic bacteria. So chlorobi, green sulfur bacteria. The electron donor is R reduced sulfur compounds. Carbon source is CO2, so they're anaerobic photolithotrophy. Purple non-sulfur bacteria. Actually, they're basically chemoheterotrophs. Now, many purple non-sulfur bacteria, they can grow pretty well in a rich medium in the dark. If they're in a very poor medium and you grow them in the light, they begin to express the proteins involved in photosynthesis. So it's, photosynthesis has been grafted onto a pre-existing type of metabolism that probably already worked, okay? So these photosynthetic bacteria could evolve from something else, okay? Because they already had a metabolism that worked. And if you add photosynthesis on, they get a competitive advantage. And then, okay, cyanobacteria, they are oxygenic photolithotropes. They manage to get electrons from water. So what was the first type of photosynthetic organism? Well, we're talking about reaction center 1 working on its own. So it's probably something like this, okay? So probably photosynthesis in eubacteria was evolved in some kind of anaerobic photolithotrophy. And the origin seems to have been in cyanobacteria, in freshwater cyanobacteria. And then, in fact, and then photosystem 1 transferred to, chlorobi. Okay, so I'm just going to go through the summary slide here. So we have different types of energy metabolism in organoheterotrophs, chemolithotrophs, and phototrophs. So for respiration, okay, the electrons originate from organic carbon. It ends up, or they end up being transferred onto reduced NADH. Then from NADH, they go along an electron transport chain, and this allows the bacteria to generate proton motive force and ATP. So kind of the transfer of energy goes along here, okay? So you have to understand what is the whole point of respiration. Well, it allows you to get 38 ATP molecules per glucose instead of just 2. That's the point. And why anaerobic respiration is not the same thing as fermentation. Okay, you need an external electron acceptor for anaerobic respiration, but not for fermentation. Chemolithotrophs, how are they living? Well, the basis of their energy metabolism starts with electron transport from some kind of inorganic mineral source. And from that, they can generate often proton motive force, and this proton motive force is going to generate both NADH by reverse electron transport and ATP. And they can fix carbon dioxide, which will be reduced by the NADH that they've managed to accumulate. So many different types, several different types of chemolithotrophs, but of course only particular combinations of electron donors and acceptors will work. So you have to remember which is the better electron donor, which is the better electron acceptor. And it determines which kind of chemistries can function. And for phototrophs, for photosynthetic eubacteria, they are polyphyletic. They do not have a common origin. And the explanation is lateral gene transfer. And it kind of makes sense because in each case, the acquisition of photosynthesis by these different groups seems to be adding an advantage onto a pre-existing type of metabolism. Okay, so it's adding on to a lithotroph or adding on to a heterotroph. So, thanks everybody for your attention. I know it hasn't been easy this afternoon because it's way too warm in this room. And that's it from me for a few weeks. So I'll only be coming back to talk about viruses later on in the semester. And don't forget, 7 p.m. I will send out a microbe of the week question. If nothing comes out, email me because I might have forgotten. Okay. Goodbye. Wow, too hot.